Okay, everyone, let us get started again on the last discussion panel. So we have the previous three speakers here in front of you, and all the questions that you were burning to ask but could not ask after their talks. Now is your chance. So the microphones are here in the front and one back here. So please come up to the microphone and ask the speakers questions. John.
Yeah, sure. So my question is for Dr. Stevens. So if you want to optimize things and minimize the cost, why would you want expansive coding? Why wouldn't you just want to sort of have compressive coding?
So, so the reason is that it's believed, this is forever, that the olfactory cortex is a big associational cortex, so that you can always find neurons that will respond to any, say, pair of odors. But in order to do that, you have to have lots of neurons that represent lots of pairs, okay, and then use those as the basis for learning to strengthen what their natural tendencies are. And so you have to have this expansion, and it's kind of wasteful, but there's no way around it because you don't know which pairs of glomeruli are going to be activated together and have that be significant, and you just have to know that.
Thank you.
Please.
This is a question for Dr. Olaf. So with connectivity with DTI, that one of the criticisms is that the fiber tracts are not real, that they're all just synthesized from these different vectors and stuff. So how can we be sure that we have real connections?
That's a really good question, and one with which people who do this kind of work struggle a lot. We're dealing with the human brain, and we're dealing with living people, and we're not going to take their skulls off and start doing dissections on them. We can't do that, and we would lose one of the great advantages of doing non-invasive imaging, which is that we can probe for behavior, we can do longitudinal studies. So there's an advantage in doing it this way. It is an inferential technique, yes, because what I've shown you as the connectome map is an inference on a complex diffusion field. It is the best fiber model that can explain the orientations of these local anisotropies that are being sampled. Having said that, we can cross-validate that map against an independently acquired imaging modality like fMRI, resting state fMRI in particular. We see many, many strong statistical relationships between these two. We can also do animal scanning, and we have done this together with Van Wedeen years ago, who has a particular expertise in imaging fixed post-mortem tissue in a high-field scanner at very high resolution, thus creating a fiber map using diffusion imaging, which we can then cross-validate again against track tracing studies, invasive histology-driven studies that have been conducted in the same species. And again, we're finding not a perfect agreement, but an encouraging degree of overlap between them. Thirdly, there are efforts underway to do post-mortem diffusion imaging in humans as well, followed by classical dissection. So it is really those kinds of approaches that will ultimately allow us to get higher confidence in the kinds of maps that we're generating. I think that the problem with diffusion imaging is not so much that the fibers we're seeing, the connections we're seeing are inaccurate, it is more that we're missing some. In that map I showed you, and I didn't dwell on too much, there are some holes where we know from classic histology, from the sexual anatomy, we should be seeing connections and we do not. And we know why we don't see them, because there are fibers passing through regions where many fibers are intersecting, and that's actually a very difficult part of the imaging process to disentangle that.
So how many connections do you think there are in the brain?
And that depends on the pacillation. That depends on how you break down the bits and pieces. In the map I've shown you, with 1,000 nodes, the filling ratio was about 3%. 3% of all possible connections actually existed, and many of them were weak. So it's actually a sparse matrix.
Lee?
So this is a question for Chuck. How would you predict which parts of the brain would have antimaps? That's one question. And the second question is, do you think there are small antimaps and large antimaps? And I mean, what's the scale and range of antimaps? So you could use structure to make maybe some predictions, but can you use function to make predictions? So I'm just curious about your thoughts.
Yes. The anatomical signature of an antimap is there has to be a group of cells that are the source and a group of cells that are the destination, and every cell in the source has to be able to make a contact with every cell in the destination. They don't have to make it, but they have to be able to make it. And so the places that I know where that is obvious are in the olfactory system, in the cerebellum, where you have the parallel fibers that all the cells that are in the beam, with all the granule cells that are in the beam, send their axons to all of the Purkinje cells in that beam. And in the hippocampus, where from the dentate to CA3, you have the mossy fibers axons that run in an arc across CA3 and have the chances to make a contact every place. So the problem that the Antimap is solving is representing all the information that's possible to have in a way so different parts of the information are emphasized more. So, for example, some odor might be emphasized in one cell just by chance, and another odor would be emphasized in another cell. And what this does is it gives you a substrate for doing learning on, so that you can put a label on any point in a high-dimensional space where you have 1,000 possible odors. That's 1,000-dimensional space, 1,000 different glomeruli, and that would be 1,000-dimensional space, because everything the brain knows about the olfactory world is represented in those 1,000 or 2,000 or whatever they are glomeruli. But that same problem happens in other places. So, for example, there's a lot of pre-processing that gets done on faces by the visual cortex. And they're going to be someplace in there, going to be a list of numbers that describe the characteristics of faces, however they do it. But to put that's going to be a high-dimensional space, but you have to be able to put, oh, there's Lee, and there's Mary Beth on those faces. And so you have to have some way of doing that. And if you did have anti-maps in the neocortex, that would be a way, but there's actually zero evidence for that. It's possible. It would be hard to see, because you'd have a bunch of cells here that are projecting all of them to a bunch over there, and they're making random connections over there in maybe a small area. So it'd be hard to detect, but it would be possible. But there may be other ways it's done.
Yes, sorry. But
I just wanted to add something. Since you have multiple ORs that are recognizing the same odorant, you have the combinations of OR inputs going into the cortex. So it's not just that Lee is represented here and Mary Beth here. It's more like Lee's foot is here and Mary Beth's hair is here, and then there are all the other parts of those, and then they come together in some cells. So I think it gives you enormous opportunity for complexity. And in fact, the way in terms of the synaptic context, it's not really known yet what's going on, although a single microcell sends projections all over the olfactory cortex, suggesting that it can connect at many different places.
I have a question to Olaf. I think it's more to you. If you compare functional connectome of the human brain and intracellular molecular network, let's say, of one cell, then my intuition would be that the number of connections is more or less similar. Some properties like rich club effect should be present in both. Some properties, let's say, of intelligence should be present also in intracellular network, like anticipation, memory, and other stuff. But still there is something different between brain and intracellular network. What properties of the network you think are different?
That's an easy question. I don't even know where to start. I mean, I might start with one commonality, which is that both cells and brains are spatial spatial systems. They are built in three-dimensional space, and somehow things, if they do interact, have to meet each other and have to connect to each other through space. And that is a major constraint for how I think cellular networks are organized, as well as neural networks are organized. This has really dawned on me really only over the past few years. I've known about it from literature for a long time, but it's never really dawned on me how much can be explained by looking at spatial embedding. So that doesn't answer your question. I don't know. I mean, for me, one of the future research directions actually is to follow sort of the networks within networks paradigm that was laid out yesterday, to see how these large-scale networks that we can now measure, perhaps incompletely and inaccurately still, but we're getting better at it. We can measure them at the whole brain level. Relate them down to the tissue underneath, the glial cells, the vasculature, the energetics of the consequences of neural signaling in terms of ATP consumption, glucose consumption. There are some very exciting relationships on the horizon. I was just telling Nathan that he talked about anaerobic glycolysis, which was mentioned yesterday also in the context of cancer. And it turns out that when you look at the regional distribution of anaerobic glycolysis in the brain, it tracks very closely the centrality of brain regions, the degree of brain regions. Highly connected brain regions use glucose very wastefully, and they almost behave like tumor cells that way. Why? Perhaps because they have to constantly turn over organelles, turn over membrane, make new components, engage in plasticity, etc. It's those kinds of relationships that I would like to explore in my future work, but that's really on the horizon.
I was wondering about where the local network neurons, the interneurons within a local region, fit in versus the projection neurons. Is there a correlation with like the hubs in your model and the short connections versus the long distance ones?
That's a good question. We have not been able to, with imaging, we cannot see local circuits. In fact, every voxel in an imaging experiment contains, on the order of hundreds of thousands, perhaps a million neurons, and that's one of the big, that's why Christoph Koch referred to it yesterday as sort of a bulk method. I think, since it allows us to look at a human brain, it has some value. We're also now imaging, working with people who are imaging and then reconstructing networks at a cellular level in Drosophila, where I think we will have a much closer shot at getting at the roles of individual neurons and cell types within the network. We have not yet been able to do that in the mouse either. The mouse connectomes that are on the horizon are also going to be mesoscopic, that is, cell regions with which have cell types in them, but we can't resolve exactly what is going on between them. So I think we're still a bit pressed for an answer here. We haven't quite gone down into the levels of detail that we need to answer the question that you've just posed, but it's an important one. Thank you.
This is a general question to all three of you. I just, because all of you actually talk about the network structure and the layout, and I was wondering, what will be your opinion how to practically speaking, how to actually study the stability as well as the redundancy of all these networks?
We have done things. Maybe that's not what you're driving at, but I think you're talking about vulnerability, maybe robustness of the network against perturbation. When you say stability, what do you mean?
Yes, I think that's one aspect of the thing, right? So actually for the two things actually I was asking about the stability and the redundancy, actually they are also related to each other.
Yes,
right?
they are.
Okay,
yes.
Because apparently the brain could actually achieve the more stable state, okay, simply because it's redundant, okay? So that, subject to certain, perturbation of the structure of the brain, actually it could actually maintain certain more stable functions, okay? But on the other hand, okay, I guess, if you want to really understand the function of the brain, okay, we need to really, go to look at, how they, from the structure point of view, how they actually really maintain this stable state and how, how to really study that. So that's why actually I try to get opinion from all of you.
I think there's a real opportunity here for network modeling, for doing systematic perturbations of networks. Once they've been acquired, once they've been mapped, we can then go in with computational methods, blow out, nodes and edges essentially, look at what the functionality of the network is afterwards, and then at the regional level that we're at now in the human brain, nothing is really redundant. Every region is unique, has its unique set of inputs and outputs, and removing any one of them will have a discernible effect on the network as a whole. If we remove regions that are more centrally embedded in the network, we see larger effects, something which we are now working with clinicians on who study stroke patients to see if there's a relationship between that and then the behavioral cognitive effects of stroke. So that would be my answer.
Yeah, I mean, I think it's an interesting question. We still have so much to learn about the brain, in terms of trying to understand these kind of things. Because I'm just thinking of sort of the hierarchy of networks. If you come down to the molecular level, which is where I work primarily, if we look at metabolic networks, for example, we can define redundancy in all kinds of ways. My first papers were actually on defining what network redundancy means, and you can do it because there's sort of a theory and you know what you're trying to explain. I think in the brain you see enormously compelling neuroplasticity, in the sense that, Mike Phelps showed some amazing data even yesterday about removal of a hemisphere, which I would previously have certainly thought that was death. And so we know that there's unbelievable plasticity in the brain in terms of rewiring and its ability to adapt. At the same time, we know that, as Olaf pointed to, certain loss of certain regions does lead massively to different loss of function. So to give a very concrete answer there, I think it's really necessary to define the scope of the problem, in terms of what networks we're sort of talking about, what networks, we were probing with what kind of, measurement tools, to really get a handle on that. Okay, thanks.
So just as a follow-up, did you try knocking out some of the nodes in this network and seeing how that affects the path length or the diameter of the graph, for example? Like sort of look at the fragility of the network.
Yes, we do that quite routinely now. Every time we get a network that we can put our hands on, we're doing those types of vulnerability studies. There are many metrics by which you can measure deficit. You can measure it by terms of path length. How does that length? You can look at things like communication processes. You can look at other things, but modularity, for instance. So we do that routinely, and the bottom line is that not all brain regions are created equal, and some are more essential for maintaining integrity of the network as a whole. Those tend to be the regions that are also most vulnerable. It looks like to metabolic oxidative stress, metabolic degeneration, and things like that. And so we're looking at that relationship in Alzheimer's disease, for example, where we can plot degenerative processes across time. Then really the network approach opens up a whole number of questions you can ask that you couldn't ask before, and that's, I think, what's so appealing about it. It has its limitations, obviously, but it allows us to ask new questions that we couldn't ask before.
Please.
All right. Well, this is kind of a two-part question for Olaf, kind of connecting back to neuroplasticity. Within healthy individuals, how much of a range do we see in the kind of rich club phenomenon in terms of where those neurons are located? Are they spatially located in certain areas consistently, or is it very widely? And additionally, have any studies been done on, say, children to see when actually those connections are laid down and when that is actually developed?
The second part, we are doing it right now, so I can't say too much about it yet. On the first part, we have not looked at individual variation too much yet. We were looking at subject cohorts of between 20 and 40 and 80 subjects. And we have looked at group averages for the most part, but quite consistent, at least in our hands, consistently, different acquisitions, different magnets, different imaging centers, other people replicating this in other places now. We see the same set of regions come out, a handful of regions that in the end are not that surprising. We've known that they are transmodal, multimodal regions that are heavily interconnected, but now we have a way of measuring how much, putting a number on it, how much are they interconnected. We can look at variations across individuals. Again, it gives us a little bit more power in our future investigations, looking at especially in individual differences, which is I think a very useful thing to do in this context.
Right. Thank you.
Question for you, Dr. Stevens.
Can you use the microphone, please?
So this was with regard to the comment Dr. Buck made about the role of interneurons here. So do you think the human-to-human variability in the way they perceive smell could be actually the role of the interneurons? So the interneurons may actually be functioning as the antimaps, allowing for that variability between. So the first question is, how do you define that it's the cerebellum and the fusiform that has the antimaps? And second, do you think it could be the interneurons that are allowing for that, species to species variability, human to human variability in the way the olfactory smells are?
So the reason for picking on the cerebellum and the hippocampus is just that they're a population of cells, all of which go to the same neurons, and every cell there gets to make can make a synapse. Doesn't necessarily, but can make a synapse with a whole population, all the same. So the cerebellum to Purkinje, the granule cell to Purkinje cells, and the dentate granule cells to CA3 neurons have that possibility of the CA dentate mossy fiber synapse runs along like that along the hippocampus, possibly making synapses with lots of cells. So that's the reason for picking those three regions. The inhibitory neurons, we don't know exactly what everything is for, but there are two large classes of inhibitory neurons in the olfactory system. In the bulb, the mitral cell axons from the bulb synapse on the layer 2 input granules. Layer 2 are called granule cells in the olfactory cortex. But there also is a feed-forward inhibition. So there are a number of inhibitory neurons that collect that same input and then feed on to the same neurons that the bulb is feeding onto. So basically, cells get excitation from the bulb, but then they also get pooled inhibition from the bulb with the feed-forward network. And this is necessary for subtracting the mean out of the signal that's coming. And that's a requirement for the compressed sensing that I didn't have time to talk about. Then there's a second set, which is a feedback inhibition, which is the same. The output of all of those layer 1 cells goes back onto layer 1 in an excitatory way, and then again the pooled output gets subtracted away, and that's subtracting the mean. So I would think that the main role of the inhibitory neurons, or at least one main role of many of them, is going to be subtracting out the mean signal so that you have plus and minus around whatever input is coming to the cells. But I'm sure there are other, when we know more about the details of the circuitry, I'm sure there are many other things that we could say are the inhibitory neurons are doing. Those are the only ones I know.
So the interneurons, you don't think could be possibly playing a role of dynamic connective matrix, if you will, that is allowing for just random connections between people to people.
As far as we know, what the inhibitory neurons are doing is they're taking the same signal that the excitatory neurons get, but then they're averaging over a whole bunch and subtracting that, so they're taking away the mean input, making the mean around 0. That's what it looks like they're for. But there are many classes of inhibitory neurons, and probably some of them are having to do with dealing with transients, temporal transients that I didn't talk about, things like that. So we really just don't know. I don't know, certainly.
Okay, last question, please, and then we need to move on. Thank you.
So this is a question for Nathan, but I think any one of you can answer. So you demonstrated in mouse that the neuronal genes are spatially separated and clearly. And in honeybee, you have shown that you can predict the behavior. So I'm kind of curious. I'm trying to find the connection between these two. One is the prediction of behavior. Another is the spatial separation. I'm just kind of wondering, is there any way that you can say or you can predict that the signals that emanate from these spatially separated neurons actually interactive, mutually exclusive, or can you develop any sort of predictive model and intuitively for human too? Thanks.
Yeah, thanks. That's a great question. And we kind of wonder, I think about this a lot because the two studies sort of have quite different results in some ways. Because in the honeybee study, right, we're able to predict behavior. That emphasizes the dynamics. Whereas in the Allen Brain Atlas, you get this immense spatial patterning, and that's across 10,000 different mice. Now, of course, they're the same strain taken at the same day, but there's not a lot of variability there. And so what I think is going to come out of this probably is that there's some studies that need to be done. We're trying actually to raise money and get some people, put together to do this. I talked to Christoph Koch about this yesterday with the Allen Brain Atlas and also with some people maybe in the context of Huntington's disease. But basically what you want is to take a subset of those genes and look at a time series analysis in the mouse where you could look spatially, at all of these things and sort of how they change dynamically. My guess is that if you were to look at, singular value decomposition or something like this, your principal modes are probably going to be defined spatially, I think. But I think in your lower degree modes, you're going to start to see changes that are related to behaviors or disease versus non-disease or whatever those kind of changes are. And so there's kind of a major need for looking at that so that we can start to see how those patterns are both the parts that are stable as well as the ones that will change dynamically in time.
Okay, thank you very much for an excellent discussion. Let's thank the speakers one more time. Okay, next, it gives me pleasure to introduce our keynote speaker, Dr.